Okay, hi everyone. My name is Jen, and I'm very glad to be here to introduce our work on disease gene prediction. And this is a joint work with Professor Yu Li, Sheng Wan, Bo Wei, and Jian Zhu. Our paper is called Disease Gene Prediction with Privileged Information and Heteroscedastic Dropout. Okay, I'll first introduce some motivation of this project. Firstly, we all know that identifying gene disease associations can help us understand pathogenesis and for searching therapeutic targets. However, experimentally verifying such a causal link between gene and disease is time-consuming and expensive. So in this project, we try to apply some computational models to find the possible gene and disease associations. And the type of model that we're going to use is graph-based models because it is one of the most popular approaches nowadays, which modeling the gene-disease relations as a link in a gene-disease network. And the most common graph-based models are like random walk-based models and some deep learning-based models. As I have mentioned before, if we model the gene and disease as a network, then we will have two kind of features. A topological feature comes from the network itself, and the gene and disease node also have their own node features. And later I will further illustrate what kind of node features we're going to use in our project. And here the model we're going to use is actually the graph neural network. Graph neural network is a deep learning model that can model these two kind of features simultaneously by message passing mechanism. And this picture briefly describes how the model works. The last motivation, I think, is the most important one. And in order to help you understand this setting, I will first introduce a scenario. Suppose now we would like to predict if a patient have a certain kind of disease or not. And we need to collect several features, including the blood pressure, blood sugar, BMI, drug history, and so forth. And after we get this data, we will feed them into the model. And at last, we have our model ready to use. For example, we can use it to make prediction for the new patients. Okay, and now let's assume we have a new patient here, and we want to predict if he or she has this kind of disease or not. So first thing we need to do is to collect the 5 features. But for some reason, this patient don't want to provide drug history, maybe because this feature is very sensitive. So what should we do in this case? Because previously the model is well trained on all those 5 features, but here we just have 4. And actually, this is very common phenomenon in bioinformatics, where features are often very difficult to collect on large scale due to technical limitations, experimental limitations, and high experimental cost. And the missing is not only restricted to the testing data, but also can appear in training data sets. And so our project is trying to deal with this kind of problems. And actually, we have 2 methods to deal with these problems nowadays. And the first one is that we can just use 4 features except drug history to retrain the model and then use this new model for prediction. Because our new model has 4 features and the new patient also has 4 features, so they perfectly match, and then we can make prediction. But it is very obvious that this will lose many useful information because we're throwing away some features. And the other method is to use original model, which take 5 features as input. And for the new patient, we just fill the missing drug history with 0 or the mean. And then we can use the previous model to make prediction for this new patient directly. And this method also has a drawback because it will introduce some bias. And if if we have a lot of missing data, the bias will more and more bias will be introduced into our model. So which will affect the model generalizability and stability. The drawback of the previous methods are what I have mentioned. We will lose many useful information. We will increase the risk of overfitting the training data sets, and it will limit the generalizability of the models. And our solution is to use a learning under privileged information framework with a graph autoencoder. And the privileged information here is actually the missing feature. And the heteroscedastic dropout is a variation of the Gaussian dropout, because Gaussian dropout has a constant variance, but for heteroscedastic dropout, we have a different variance for each node. So to be simple, that is to say, the variance is varying, it's not a constant. And now I'm going to talk about the idea of our model. We model the gene and disease as a heterogeneous network, as shown here. We have different kind of relations defined by the nodes. So it is a heterogeneous network. And our assumption is that similar genes are more likely to be associated with a similar set of diseases. And similar diseases tend to share similar disease genes. So this is a very basic assumption of our project. And this slide tells you about the relationship between dropout and the privileged features or the missing features. Because we all know that dropout is a widely used techniques in deep learning. It is only used in training phase, and after model is well trained, it will be discarded. So in our setting, there are some features, the missing feature only appear in the training data set, but not the testing data set. So this is a this perfectly coincide with each other, right? So why not just put those missing features in the dropout and use it to help train the model instead of discarding them away. And in testing phase, we don't need it anymore. So this is a very important idea for our model. This is a simple paradigm of our model where we use one model taking the regular feature as an input to control the mean, and the other model taking missing feature as input to control the variance of this Gaussian dropout. And then we combine them together to generate a joint learned embedding of the nodes. Basically, we just combine them in a fully connected layer to output this H plus, this joint learned embedding for that node. And why we need to do this is because first, we need to control the variance of the learned embedding, because many genes only have some features in the feature set, and the missing features will be filled with 0 or the mean. And thus, a lot of bias will be introduced, which will lead to unstable results. And the second, we don't want to throw away those valuable information. So in this case, we introduce Gaussian dropout in this model, where mean is determined by the main model, taking the regular feature as input. And we also introduce another dropout model to determine the variance of the Gaussian dropout, taking the privileged features as input. Okay, and as I have mentioned before, the graph-based model we use here is actually the graph neural network, and the propagation rule is defined like this. However, the graph we use here is a heterogeneous network, which means it has 3 types of relations, namely the relations between gene and gene, between gene and disease, and between disease and disease. But the graph convolutional neural network only model one type of relationship. So here the model is a variation of the graph neural network. It is called relational graph convolutional neural network, which can model multiple relations. So we can see from this equation that the capital R here is a number of relations that we would like to model, and WR here as a weight matrix that introduce multiple message passing mechanisms. And the joint embedding we learned comes from the mainnet and the dropout net. And the mainnet control the mean and the dropout net control the variance. And we also have a variance sharing version to avoid overfitting. So we train our model via variational graph autoencoders. And our objective function is just a variational lower bound shown like this. And the QZ here is actually the conditional probability, conditional distribution of the latent representation in low dimension given the input. And the PZ here is actually the prior distribution on this latent representation. And we can see from here that the first term actually measures entropy loss, and the second term is a regularization term. So this equation as a whole seeks a trade-off between model fit and the model generalizability. And at last, suppose we have two learned embeddings, MXI and MXJ of two nodes. Then we apply an inner product on these two embeddings, and then apply a sigmoid function to output a probability to determine if there is a link between these two nodes. So this is a very simple decoder. Okay, next part, I will going to talk about the data sets we're going to use and also the experiment. The data sets or the network are constructed from some well-known data sets. And the gene features include the gene expression function associations from different species. And the disease features include the ontology and annotations from the human disease ontology, and also some clinical features. So for the experiment, we first measure the overall performance of gene disease prediction. And we can see from this result that the LUPI-RGCN performs better in every settings. And we also consider the performance of our model on other settings. And here we consider three situations: singleton genes, new diseases, and novel associations. So basically, singleton genes means a gene that has only one association to the other diseases. And we want to see if we can recover it if we remove it from the training data set. And disease as the disease, a new disease is kind of disease whose adds to other genes will be removed. And we also would like to see if we can recover them in testing phase. And novel association is actually the most difficult task here. It is referred to the type of gene disease associations where we didn't see either of those two nodes in the training data set. And we also want to see if we can recover it in testing phase. We can see that our model LUPI-RGCN performs better than in other settings. So in this project, we proposed a learnable heteroscedastic dropout where mean and variance are determined by two RGCNs respectively to deal with a missing feature situation. And our computational framework, I think, is very general learning paradigm that could be easily adapted to other biological applications where collecting certain kind of features is very difficult. And also, we are very interested in interpretable deep learning. So in the future work, we might focus on generalizing some interpretation framework to interpret our model predictions. And that is all for the brief introduction of our projects. Thank you.